Have you ever wondered how our digestive system works? What happens to our food after we eat them?
The food undergoes the process of digestion. Digestion is the breakdown of large insoluble food compounds into small water-soluble components so that they can be absorbed into the blood plasma and then can be utilized for energy and growth. Digestion is a mechanical and chemical process that begins in the mouth and ends at anus. Our food's journey begins in the mouth. Mouth is a horizontal transverse slit-like aperture which is surrounded by upper lip and lower lip. A special muscle is associated with lip called orbicularis oris muscle. Saliva in the mouth contains salivary amylase, an enzyme that begins the breakdown of starch into simpler sugars like maltose. This early stage of carbohydrate digestion helps prepare the food for further breakdown in the stomach and small intestine. Saliva also moistens our food, making it easier to swallow. Next stop is the esophagus. It is a hollow tube that connects our mouth to our stomach. After we swallow, our food travels down this tube as it encounters powerful squeezes and squishes. The movement of the esophagus is called peristalsis. Tension and relaxation of the muscles in the esophagus that pushes food down into the stomach. Now, we're in the stomach, a stretchy bag that squishes and mixes the food with stomach juices. The stomach's primary function is to digest food by both mechanical and chemical means, breaking it down into a liquid mixture called chyme, then passing it into small intestine. It also temporarily stores food after swallowing. Chemical digestion in stomach. Bolus, a small rounded mass of chewed up food, enters the stomach through the esophagus via the lower esophageal sphincter. The stomach releases proteases, protein-digesting enzymes such as pepsin and hydrochloric acid, which kills or inhibits bacteria and provides the acidic pH of 2 for the proteases to work. Food is churned by the stomach through peristaltic muscular contractions of the wall, reducing the volume of the bolus and the body of stomach as the boluses are converted into chyme, partially digested food. Chyme slowly passes through the pyloric sphincter and into the duodenum of the small intestine, where the extraction of nutrients begins. Gastric juice in the stomach contains pepsinogen. Hydrochloric acid activates this inactive form of enzyme into the active form, pepsin. Pepsin breaks down proteins into polypeptides. Acidity of stomach inactivates salivary amylase and activates lingual lipase. Lingual lipase then begins breaking down triglycerides into free fatty acids and mono and diglycerides. The breakdown of protein begins in the stomach through the actions of hydrochloric acid and the enzyme pepsin. The stomach can also produce gastric lipase, which can help digesting fat. Absorption in stomach. Absorption of certain small molecules occur in the stomach through its lining. This includes water, if the body is dehydrated, medication, such as aspirin, amino acids, 10 to 20% of ingested ethanol, e.g. from alcoholic beverages, caffeine. To a small extent water-soluble vitamins, most are absorbed in the small intestine. The parietal cells of the human stomach are responsible for producing intrinsic factor, which is necessary for the absorption of vitamin B12. B12 is used in cellular metabolism and is necessary for the production of red blood cells and the functioning of the nervous system. As the ball-like mixture turns into a frothy liquid, it is now ready to move into the intestine. The intestine has two sections, the small intestine and the big intestine. Our food gets broken down even more in the small intestine, and our body takes all the nutrients from it. The small intestine is about 5.5 meters, 18 feet, long and folds many times to fit in the abdomen. Although it is longer than the large intestine, it is called the small intestine because it is narrower in diameter. The small intestine is divided into three structural parts. The duodenum is a short structure ranging from 8 to 10 inches in length and shaped like a C. It surrounds the head of the pancreas. It receives gastric chyme from the stomach, together with digestive juices from the pancreas and the liver. The digestive enzymes break down proteins and bile emulsifies fats into micelles. The duodenum contains Brunner's glands, which produce a mucus-rich alkaline secretion containing bicarbonate. These secretions, in combination with bicarbonate from the pancreas, neutralize the stomach acids contained in gastric chyme. The jejunum is the midsection of the small intestine, connecting the duodenum to the ileum. It is about 2.5 meter long and contains the circular folds and intestinal villi that increase its surface area. Products of digestion are absorbed into the bloodstream here. The ileum is the final section of the small intestine. It is about 3 meter long and contains villi similar to the jejunum. It absorbs mainly vitamin B12 and bile acids, as well as any other remaining nutrients. The ileum joins to the cecum of the large intestine at the ileocecal junction. Small intestine. The small intestine is where most chemical digestion takes place. Many of the digestive enzymes that act in the small intestine are secreted by the pancreas and liver and enter the small intestine via the pancreatic duct. Pancreatic enzymes and bile from the gallbladder enter the small intestine in response to the hormone cholecystokinin, which is produced in the response to the presence of nutrients. Proteins are degraded into small peptides and amino acids before absorption. Chemical breakdown begins in the stomach and continues in the small intestine. Proteolytic enzymes, including trypsin, activated by enterokinase, and chymotrypsin, are secreted by the pancreas and cleave proteins into smaller peptides. Carboxypeptidase, which is a pancreatic brush border enzyme that remove the terminal amino acids from the carboxy end of a peptide or protein chain. Aminopeptidases are enzymes that break down proteins and peptides by cleaving amino acids from the N-terminus, and dipeptidase free the end amino acid product. Pancreatic lipase breaks down triglycerides into free fatty acids and monoglycerides. Pancreatic lipase works with the help of the salts from the bile secreted by the liver and stored in the gallbladder. Bile salts attach to triglycerides to help emulsify them, which aids access by pancreatic lipase. This occurs because the lipase is water-soluble, but the fatty triglycerides are hydrophobic and tend to orient towards each other and away from the watery intestinal surroundings. The bile salts emulsify the triglycerides in the watery surroundings until the lipase can break them into the smaller components that are able to enter the villi for absorption. Some carbohydrates are degraded into simple sugars, or monosaccharides, e.g., glucose. Pancreatic amylase breaks down some carbohydrates, notably starch, into oligosaccharides. Other carbohydrates pass undigested into the large intestine for further handling by intestinal bacteria. Brush border enzymes take over from there. The most important brush border enzymes are dextrinase and glucoamylase, which further break down oligosaccharides. Other brush border enzymes are maltase, sucrase, and lactase. Absorption in small intestine. Digested food is now able to pass into the blood vessels in the wall of the intestine through either diffusion or active transport. The small intestine is the site where most of the nutrients from ingested food are absorbed. The inner wall, or mucosa, of the small intestine is lined with intestinal epithelium, a simple columnar epithelium. Structurally, the mucosa is covered in wrinkles or flaps called circular folds, which are considered permanent features in the mucosa. From the circular folds project microscopic finger-like pieces of tissue called villi, Latin for shaggy hair. The individual epithelial cells also have finger-like projections known as microvilli. The functions of the circular folds, the villi, and the microvilli are to increase the amount of surface area available for the absorption of nutrients, and to limit the loss of said nutrients to intestinal fauna. Each villus has a network of capillaries and fine lymphatic vessels called lacteals close to its surface. The epithelial cells of the villi transport nutrients from the lumen of the intestine into these capillaries, amino acids and carbohydrates, and lacteals, lipids. The absorbed substances are transported via the blood vessels to different organs of the body. The material that remains undigested and unabsorbed passes into the large intestine. Absorption of the majority of nutrients takes place in the jejunum, with the following notable exceptions. Iron is absorbed in the duodenum. Folate, vitamin B9, is absorbed in the duodenum and jejunum. Vitamin B12 and bile salts are absorbed in the terminal ileum. Vitamin B12 will only be absorbed by the ileum after binding to a protein known as intrinsic factor. Water is absorbed by osmosis and lipids by passive diffusion throughout the small intestine. Sodium bicarbonate is absorbed by active transport. Fructose is absorbed by facilitated diffusion. The large intestine then takes out all the water from what was left of our food. The result is a solid mass called stool. The stool enters the rectum. It is the last section of the large intestine. It holds the formed feces awaiting elimination via defecation through anus. It is about 12 centimeters long. In mammals, the large intestine consists of the cecum, including the appendix, colon, the longest part, rectum, and anal canal. The four sections of the colon are the ascending colon, transverse colon, descending colon, and sigmoid colon. These sections turn at the colic flexures.